Hi everyone, and welcome back to Neuropsychology. So in this video we are going to cover some methods that we use to study the brain, or methods used in neuroscience. So we're going to talk about single unit recordings, optogenetics, and some dynamic brain imaging methods such as PET scanning or MRI scanning. So let's get started. So there are many methods we use to study the brain and behavior. You must have seen these plots before, where the temporal resolution of various methods of probing brain function is plotted along one axis and their spatial resolution on the other. So spatial resolution is often approximated in terms of units of the nervous system. So for example, it starts off of dendritic spines all the way to cortical columns or even lobes and hemispheres. So similarly, temporal resolution is indicated with labels such as milliseconds or hours and beyond. So in this plot, functional MRI or fMRI, which can resolve brain activity down to millimeters into seconds, occupies at a different position on the plot than, for example, EEG, whose temporal resolution is much more accurate within milliseconds, but the spatial resolution is not as accurate. So the techniques that boast the highest resolution in both space and time are generally more invasive. So an example of that is intracerebral microelectrode arrays. However, even though these microelectrodes can resolve individual neurons or can measure spiking of individual neurons, it is practically impossible to record for more than a few small patches of brain at a time. Whereas, for example, with fMRI, you can look at the whole brain at once. So we're going to talk about a couple of these methods, including PET scanning, EEG, single unit recordings, fMRI, just to name a few. Okay, so single unit recordings provide a way of measuring the electrophysiological response of a single neuron, or maybe two. In this method, research use a tiny little microelectrode system. So when a neuron generates an action potential, the signal propagates down to the neuron, and then this electrode can measure this action potential. So a microelectrode can be inserted into the brain wherever you want to measure it, and it can record the rate of change of voltage with respect to time. So these microelectrodes must be super fine tipped and have high impedance conductors, and they're mainly made of glass micropipettes or even little metal microelectrodes made of platinum or tungsten metal. So this procedure is super invasive, and we use animal models for this. So we also study this at FIU. We also use this method at FIU for research to measure neuron spikes. So single unit recordings are super localized and very precise. So you have to calculate exactly where you want to put these electrodes in and how far to drive them down into the brain. So after recording the spiking of the neurons, and when the experiment has finished for that specific animal, the animal will be sacrificed and the brain can be extracted, so taken out. So then the brain can be sliced in really tiny thin slices, and with the help of a microscope we can see exactly where these recordings took place, kind of as a final check. So another method that's actually really cool is optogenetics. Optogenetics is a technique that involves the use of light to control cells in living tissue. So, for example, in the brain, in neurons. So these neurons have been genetically modified to express light-sensitive ion channels. So this light-sensitive ion channel, which is called channel rhodopsin, opens up in response to light. So this ion channel is technically similar to what we talked about in the past when we talked about these proteins that could be ion channels to let ions in or out. So this protein can be activated when shining a light on it, which will generate an action potential within the cell. Scientists have been able to create rodents that naturally express this protein, or it can also be injected into the brain, into a very specific brain area. So then you can shine the light to turn these cells on or off, and you can even do this in freely moving animals. So researchers can precisely measure these manipulation effects in real time. So often this is done whenever the rat is doing a behavioral task, and then with this light they turn brain areas on or off to see how that affects their performance on that behavioral task. Researchers can therefore target specific brain areas and then again turn them on or off to see how it changes the behavior of the animal. So here you see a very schematic picture of optogenetics with the 6 steps. So you have in step 1 how you piece together the genetic construct. In step 2 that the genetics are inserted into a virus. In step 3 the virus is injected into the animal brain. So opsin will be expressed into the targeted neurons. So you have to be super precise here in what brain area you want to inject this in. And then in step 4, you insert the optrode or a fiber optic cable plus the electrode. And in step 5, you can turn it on or off, and this will open up these channels to let action potentials, to start action potentials in the cell. Okay, so we talked about 2 animal procedures. Let's talk about something we use in humans. So in humans, we use dynamic brain imaging. So when a brain region is active, the amount of blood, oxygen, and glucose flowing into that region increases. So we can measure the change of blood flow and oxygen in the brain using a bunch of different methods. So using these measures, we can infer from these results changes in brain activity. So the methods that are developed based on this are, for example, PET or positron emission tomography, functional magnetic resonance imaging, and also something called optical tomography, but we're not going to talk about the last one. Okay, so let's start off with PET. So when studying positron emission tomography, or PET, researchers study metabolic activity in brain cells that are engaged in processing brain functions such as memory or language. So PET technically indirectly detects changes in the blood flow. The PET scanner is kind of like a donut-shaped array of radiation detectors that encircle a person's head. And a small amount of water labeled with radioactive molecules can be injected into the bloodstream, often in the arm. And then these radioactive molecules will go into the brain, and then eventually they will break down within a few minutes, and they are quickly eliminated from the body, so it's not dangerous at all. So when these radioactive atoms give off their radioactivity, they technically become plain old regular atoms again. So when the whenever the radioactivity gets given off, so whenever it lowers in radioactivity, it gives off a subatomic particle called a positron. So again, these cells, these radioactive molecules will eventually break down, and then they will give off a positron. So a positron is a tiny positively charged particle. And whenever this positron hits an electron at the right speed, they will combine and technically destroy each other. So the energy that is released when these two hit each other, an electron and a positron, will be released as 2 gamma rays that will travel straight out of the patient's body in opposite directions. So the PET scanner can detect these gamma rays and then detect where the tracer is exactly in the body. And by detecting many of these per second, and of course we need some clever computer processing, we can look at brain activity in multiple dimensions. So here you see another picture of it. Here you see a PET scanner. Here you see a schematic PET scanner, and this is a picture you can create with that. So a small amount of radioactively labeled water is injected into the subject, active areas of the brain use more blood and thus have more radioactive labels. And then positrons from the radioactivity are released, they collide with electrodes in the brain, and photons are produced, exit the head, and are detected. So this is kind of a recap of everything I just said. Okay, so here you have another schematic photo of it, because I know it gets a little bit confusing, so I keep repeating myself, but I think that's good. So again, whenever a positron is released by the unstable nucleus of the radioactive molecule, so here you see the molecule, the positron is released. Whenever it hits an electron, these two gamma waves will go out of the body in opposite directions. Then the coil can measure that, and then with all the different slices, we can measure at all these different slices. Okay, so in modern PET cameras obtain multiple parallel brain slices at the same time. So images consist of voxels, which are technically like pixels, but they're 3D images that are about 2 by 2 by 2 millimeters. And PET does not actually measure neuroactivity. It infers by assuming that blood flow increases in areas where neuroactivity increases. Okay, so one thing I did want to note before moving on to the next one is that positron emission tomography, or PET, uses a subtraction method. So it shows a difference in blood flow between two states. So you have the blood flow pattern during an experiment minus a blood flow pattern during rest. And then you can see how much higher the blood flow pattern is during the experiment. So PET has many advantages. So the advantages of PET are it can detect a wide range of radiochemicals. It can also detect changes in your pH, glucose levels, amino acid levels, and proteins. It can detect relative amounts of a neurotransmitter, density of receptors, or even degenerative processes that might be related to diseases or things such as typical aging. And it can be used to study cognitive functions such as language. So the disadvantages are the measurement of the neuroactivity is not direct, so we often have to make assumptions or infer something from the data that we get, and it uses a subtraction process, so we cannot directly measure something. Okay, so the next method is MRI. MRI stands for magnetic resonance imaging. And in MRI, a large magnet and certain radio frequency pulses generate a brain signal that produces an image. So MRI is used to study both brain anatomy and neural function, and it's very non-invasive. So it's safe enough to use repeatedly on volunteers and also on patients. It's technically just a big magnet. So there are magnets with different magnetic fields, and we measure this in Tesla. So often the scanners are between 1.5 and 7 Tesla, for humans at least. And at FIU here, we have a 3T MRI scanner in PG5. So this is what the scanner looks like. It is not very old. We just got it a couple years ago. And this is when it was inserted into the building. It's very, very heavy and a very, very expensive machine. Okay, so the patient or subject is technically placed in a long metal cylinder and has 2 sets of magnetic coils. So technically what this picture is missing is you see the big MRI scanner and you see the bed they're lying on and you see the magnetic coils right here. But it is missing a radio frequency coil, which normally goes right around the head, just like a football helmet. So it kind of works like a big camera by interrupting the magnetic field to produce an MRI image. Okay, so let's dive a little deeper into the physics behind magnetic resonance imaging. So MRI is based on the principle that our body and brain consist of soft tissue, which contains water, and water contains hydrogen atoms. So these hydrogen atoms, they have a nucleus with a proton, and each proton spins or rotates around its axis, and it technically acts as a small magnet because it has its own north and south dipole. So each spinning proton produces a tiny electrical current. So normally these proteins are oriented at random. So a given piece of tissue has no net dipole and therefore does also not have a net electrical current. So you can see that right here in picture A. So when we place these spinning protons or the soft tissue into a magnetic field such as an MRI scanner, the spinning protons orient themselves with respect to the field line of force. So you can see that right here. So in the beginning they were at random and then they all line up with the magnetic field. So the protons kind of act like a compass needle and aligns itself north and south with the Earth's magnetic field. When they are aligned like that, the summed electrical current of these protons is big enough to be measured. Because the density of these protons is different in different brain tissues, so for example it's different in CSF or in myelin or even in neurons, the electrical currents that are produced by aligning these protons is also different. So it's higher for some tissues and it's lower for other tissues. So measures of this electrical current are technically what creates these MRI images. It's cool, huh? It's pretty complex. But it does not stop there. So that's not all. So when we have a brief radio frequency pulse that comes from the football helmet I just talked about, when that brief radio frequency pulse is applied to the brain, perpendicular to the magnetic field, this technically pushes these protons on their side. So if you remember from the previous picture, the proton was first here, spinning, and then this radio frequency pulse kind of just pushes it out on his side. So now they have two motions. These protons will spin around their own axis, and they will also wobble like a spinning top. So this movement is something that we call precession. So this precession movement produces magnetic field vertically and also horizontally. So when this horizontal radio frequency pulse is then again turned off, these protons relax and they will move back towards their initial alignment. During this relaxation, we can measure the difference in the magnetic field. So protons in different tissue types have way different relaxation rates. So for example, CSF or cerebrospinal fluid has a way slower relaxation rate compared to brain tissue, for example. So we can translate these different measurements into brain image gradients. So some will be darker and some will be lighter. So for example, in a T1 weighted image, fluids are darker and brain tissue is lighter. In another type of image, which we call T2 or T2 weighted images, fluids are lighter and the brain tissue is darker. So another MRI method that is often used is called diffusion weighted imaging or DWI. Diffusion weighted imaging detects the directional movement of water molecules to create images of the brain fiber pathways. So as we just said, soft tissue is everywhere in your brain, and soft tissue contains water molecules. So for example, water molecules in the ventricles and even in cell bodies are more relatively unimpeded in random directions, so they kind of move around. However, in fibers, their movement is restricted by the track's orientation, and its contents, it tends to follow the direction of its longitudinal axis. So for example here, this could be in CSF, where these water molecules just randomly move around. And here, if you have these long axons with the myelin around it, which is a fatty substance and water cannot go through it, the water will go along with these axons. So technically what we call this is this restricted movement of water molecules along the longitudinal axis of fibers is what we call anisotropy. So we can use this method to investigate brain pathways and also connectivity. So here's a really cool picture where people used tractography to follow these water molecules and see what the pathways in the brain look like. Looks cool, right? And all these different colors could either be different pathways or they could be different directions. So here you see a schematic picture. This is from the book. This is a pathway between Broca and Wernicke's area, which is very important for speech. And this is created with DTI tractography. So here every pathway is a different color. So you see technically 3 pathways between Wernicke's area all the way to Broca's area. Okay, so next we are moving on to fMRI. So fMRI stands for functional magnetic resonance imaging. So what I want you to know is as neurons become active, they use more oxygen, which will result into a temporary dip in the amount of oxygen in the blood. So at the same time, active neurons signal the blood vessels to dilate and increase blood flow so more oxygen can go to that brain area. So when human brain activity increases, there will be an increase in blood flow and therefore an increase in oxygen. This increase alters the magnetic properties of water in the blood, so technically it is measurable. Functional magnetic resonance imaging, or fMRI, can accurately match these changes in magnetic properties to specific locations in the brain. So before neuroactivation, the amount of deoxyhemoglobin, or hemoglobin, blood cells without oxygen, and oxyhemoglobin, or hemoglobin with oxygen, are about equal. However, after neuronal activation, the amount of oxyhemoglobin in the blood increases. So the magnetic properties of unoxygenated blood are higher than those of oxygenated blood. So let's say, for example, when you're lying in a scanner and you get an fMRI done, when you get a visual stimulus, so for example, you have to watch a movie, during an fMRI test, there will be a lot of oxygenated blood going to your visual cortex or towards the back of the brain. So if you put this fMRI picture on an anatomical image, you can see exactly where in the brain there was an increase of activation. Okay, so here's a little recap. So whenever neurons are active, they need more oxygen. So oxygenated blood will go to that area. So there will be an increased amount of oxygenated blood. And then we can measure the difference of the unoxygenated blood and oxygenated blood to see where there is an increase of activation. And activity patterns in the MRI represent higher levels of oxygenated blood in that brain area. So even an easier recap, if there is more oxygenated blood, there is more activity. So there are a lot of advantages of fMRI. You can see changes in neuroactivity. There's no subtraction process, although sometimes we do use that for analysis. It's high spatial resolution. You don't have to average across subjects if you don't want to, and it's not invasive at all. The disadvantage is that the temporal resolution is not great, and MRIs are crazy expensive to get, especially in hospitals. And even though they're not harmful, there are loud noises, so sometimes people are not very comfortable getting them. So this was the end of this video, and I will see you guys back soon for the next video.